few words with you all, and I just want to say a huge kudos to the societies for setting up a really superb schedule of events, except for this bit where you've had me. But other than that, absolutely brilliant lineup for the whole week. So it's really great to see this much interest. Thank you for the invite, and I'm going to focus on the issue of health inequalities and health disadvantage. And this is an area that I'm particularly interested in. And it has a particular issue now in the context of COVID. So what my plan is to look at the concept of health inequalities, look at the evidence that we know of within the UK and Ireland and the EU and more worldwide. And I'm going to touch on the social determinants of health and what they are and what that means for us, and then look at that within the context of COVID. So that's my plan. I know when I was asked to speak, I was asked to maybe refer to working on in the clinical area during one of the most recent surges. But to be honest, I think it wouldn't be right for me to do that. I'm not a frontline worker. The nurses and midwives out in practice at the moment are the ones who are frontline workers. So I'm going to focus on what I do know a bit more about, and that's what I'm going to look at today. So health inequalities, and we just to define what we're talking about when we talk about health inequalities is the fact that we know that people who experience material disadvantage, lower education opportunities, lower opportunities in employment are more likely to experience worse health than the rest of the population. We know as well from lots of evidence that people who live in materially deprived neighbourhoods can be seen to have poorer health than other areas. So when we think about health inequalities, it's a very large sort of concept, but what exactly are we talking about? We could be talking about inequality in terms of mortality rates, in terms of survival of particular conditions. We could be talking about inequality of morbidity and diseases, inequalities of health outcomes, of access to services is often referred to as an issue of inequality. We also know of inequalities in terms of self-perceived health. So even when we ask different people in different communities about their health, their own self-perceived ideas of health are inequitable. We also know that there's inequalities experienced by different groups, and that is often referred to in the sociological literature as intersectionality, and I'll come back to that a little bit later on. And we also know of an inequality within populations and between populations when we compare different populations. And for many of you, you might have come across some of this evidence before. Sadly, it's not new. We know how we know evidence and statistics from many locations. I've just realized I've made this view very small for myself because I put it in presenter view, so I can't quite see this slide, but I know what's on it because I've seen it so many times. So this is coming from the UK, this evidence. And can I just check, by the way, that you're seeing just the full slide on the full screen? Yeah, you're not seeing exactly what I'm seeing. Okay, that's good. Sorry, I'm down to one screen. I'm so used to two. Okay, so what you've got here is the life expectancy going from most deprived areas in the UK across to the least deprived areas, over to the right, to your right of the graph, the least deprived areas. So those are the more wealthy parts of the UK, the more deprived areas down on the left. And you can see now the different colours of green aren't too important. They're different parts of the UK. But what is important about them is that they're all mapping in the same direction. So even in really well-off parts of West London or parts of the UK that might be considered well off, that you have this trend that life expectancy is lower in most deprived areas. When you compare, when you look at that, look at the distinction that there is between the most deprived area and the least deprived area in terms of life expectancy. This is disability, sorry, I should be clear, disability-free life expectancy. So this is number of years that people are expected to live without a disability impeding their life. So it's a really stark differential in life expectancy or disability-free years. Let's look at another bit of evidence. This is coming from Spain, and this is to do with mental health. Mental health among workers, and the different shades of blue indicate different types of security of employment, if you like. So the darker blue is permanent workers. The lighter blue, then, or the next shade of blue, is the fixed term temporary. I don't know, I could actually just use my little laser pointer, couldn't I? Can you see that? So the fixed term temporary and the non-fixed term temporary. And this is the percentage of people who reported poor mental health. So they're all manual workers. They're all in Spain. The distinction is between the type of contract they have. So there's a differences, of course, between men and women to be seen as well. But even just putting that aside for one moment, you do see that the people with no contract, so they don't know if they've got work next week or next month, and the people who are permanent, there's a vast difference in mental health difficulties between them. There is a difference between men and women, which is interesting as well, and we could talk about that another day. And I enjoy using this slide in class, and I ask students to speculate as to why it might be that women, even though they've got better security of tenure than some of the men, they still have poorer mental health. But that's another discussion for another day. But interesting to see that we've got employment having a big impact on mental health issues. Let's look a bit closer to home then in Ireland. This is evidence from the CSO, the Central Statistics Office. And very clearly, sorry, this is a little bit too small to see probably, but very clearly persons in the most deprived areas have lowest life expectancy. And these are mortality differentials that are fairly stark here in Ireland. All of this data is very useful for you in terms of writing assignments or researching this issue. So I'd encourage you to use the CSO. It's a really superb source of evidence and data. Another point is suicide and mental health. We know as well from studies that across the island of Ireland, that the rates of mental ill health and suicide are significantly higher in economically deprived areas. For example, a study in Northern Ireland showed the average suicide rate in economically deprived areas to be twice that of non-deprived areas. The rate of hospitalization for mental illness also is highest among the unskilled group. Women in the low socioeconomic group in Northern Ireland were 60% more likely to experience some form of a neurotic disorder than those in the highest. So these are fairly stark differences. And this is an issue that affects across the lifespan. We know that in older age, older people living in poverty and isolation are at greater risk of ill health, and they often have this in isolation, and even more so now with COVID, and isolation is a major issue, but they also struggle to make their needs known, even though they're often those in the people that are in most need of support. The other end of the age spectrum, in terms of children, from the cradle to the grave, we know that inequalities in health impact. Children born in less well-off areas are more likely to die before the age of 1 year than those born in the most advantaged areas. And this is a stat from 1999 that was used in the evidence from the Marmot review, which I'll come to in a second, showed that the perinatal mortality rate, when it was looked at with the father's occupation, was 3 times higher for children of unskilled manual workers than for those born in the higher professional categories. And there's evidence around low birth weight babies and childhood accidents as well in terms of socioeconomic status. Back to Ireland again, and very often we pride ourselves in Ireland with very high quality cancer care in Ireland, and that is the case. But there is an inequality in cancer as well in Ireland, and this is some pieces taken from a report from the Irish Cancer Society. I think last year, possibly the year before, when they were celebrating their 50 years of the Irish Cancer Society. And in their report, they said that despite the strides made in cancer diagnosis and treatment, Ireland has become a very unequal society when it comes to health problems, particularly cancer and access to healthcare. There's a worrying divide between, there is a worrying divide emerging, and this is a challenge that faces all of us, the policymakers, healthcare providers. The Irish Cancer Society said that we must work now to begin to close this gap, and closing the gap was a phrase that was used previously by Sir Michael Marmot in relation to social determinants of health. So we know that the data around cancer is in terms of both cancer rates and cancer survival, that there is distinction between people in disadvantaged communities. And we also know that the related deaths of cancer are double those that in the highest occupational classes. Little bit more on cancer now, because I think it's very important. We know, of course, that cigarette smoking accounts for up to half of the differences in life expectancy, but we know that it's also, these cancers are also more common in areas of higher unemployment and deprivation. The bad news about this is none of this is new. We've known about an awful lot of this for some time. This is a very important report that was published, but in 2008. In 2008, the Combat Poverty Agency, who have since been disbanded, they published this report looking at tackling health inequalities, looking for an all-Ireland approach to the social determinants of health. And they said, and this is a really important quote, they said that although public patients tend to be older, sicker, and poorer than private patients, there has been evidence of lengthier waits for treatments for those who rely on public hospital services. That was in 2008. And we all know that the issues in relation to waiting lists in Ireland, and we know as well that COVID has particularly made that situation even worse again. So this quote, I suppose, points straight to the issue of the health service around public versus private. Let's look wider then to Europe. We know of health inequalities in Europe. This is a report the task were involved in around health inequalities, and it really highlighted the issue of inequalities in Ireland. If you get a chance to read this report, if this is something you're interested in, I recommend you go to the foreword. It's got some really useful evidence. But it does say that health inequalities in Europe, and you can literally search for the word Ireland through it and see where we rank, and it's not great. Within European countries, though, we know there are stark social gradients in morbidity, mortality, and life expectancy. And the higher the social position, and that's usually measured by levels of education and employment or position on the income distribution, then the better the health of those individuals in that population. Oh yeah, sorry, that's a link to another piece of research within Europe, the European Commission. Sorry, I actually thought I had deleted that slide, apologies. So there is just a little bit more information there from a European perspective. The World Health Organization then has also had concerns around health inequalities, looking at the global perspective. And you can imagine that in poorer countries, health inequities are even more starkly differentiated. Health inequalities, they define them as differences in health status or in the distribution of health determinants between different population groups. Differences in mobility of elderly people and younger populations. So they also go on to distinguish between inequality and inequity, and that's an interesting distinction to look at. And they say that some health inequalities are attributable to biological variations or free choice. I'm not too comfortable with that, that bit of this quote, but others are attributable to the external environment and conditions mainly outside the control of the individuals concerned. It's a very politically correct way of saying that some people are in situations that they can do very little about, but yet their health is incredibly impacted and poorer. All of you in public health will have seen this diagram numerous times and will have been encouraged to consider the wider socioeconomic inequalities, which you can see on the outside of this diagram, the general socioeconomic, cultural, and environmental conditions, that these all impact on ultimately on each individual person, but also on their communities. Now, in a lot of the time within fairly emergency scenarios, in a lot of healthcare, we tend to focus on the individual lifestyle factors. We tend to focus on diet, we focus on cigarette smoking, substance use, healthy lifestyle. And what's important, of course, is that while we might encourage our patients and clients to improve these, there is very little they can do about most of the others, which are impacting quite significantly on their lives, which they can do very, very little about. So there's a real risk when we focus too much on the individual lifestyle factors that we blame individuals for circumstances that they can do nothing about. How am I doing for time, Kate? Am I kind of halfway through?
Yeah, Angela, maybe just a little bit past it, yeah.
Okay, perfect, thanks. So just another little bit of lovely piece of bedside reading I would recommend for anyone who hasn't read it already, is The Spirit Level by Wilkinson and Pickett. And what was interesting about this book was that not only did they, now I know it has its critics in terms of some of the methodologies they used in their analyses, but what it shows is that in countries where there are inequalities, so in more unequal a country is, the greater that country, the whole country, will experience other issues of enduring levels of material deprivation. So that the more inequitable a country is, the higher the rates of juvenile delinquency, of substance misuse, of youth imprisonment, of teenage pregnancies, so they map page after page showing the differences between countries that are more equitable and countries that are less so. Really worth reading and very easily digested. So I've gone from health inequalities to looking at the social determinants of health. We tend to focus on healthcare systems a lot as well, don't we? Those of us who work in hospitals and in healthcare. But what's interesting to know is that even in a country like the UK, which this report relates to, even in a country with a health system which is free at the point of delivery, the NHS, often held up as a wonderful example of healthcare. It is free at the point of delivery, but even there, and I won't click on the link because I'll let you look at it if you want to, this report tells us that there are stark inequalities despite the health system. And that's very interesting to note. So those of you who are interested in this area will have heard of Sir Michael Marmot. And in 2008, he was commissioned by the then Secretary of State for Health in the UK to chair an independent review to propose the most effective evidence-based approach to reducing health inequalities in England. And the final report was called Fair Society, Healthy Lives, and it was published in 2010. And what's interesting is these are the key actions he came up with, or they came up with. It was a huge piece of work, and based in the Centre for Health Equity, I think it's called, in UCL, University College London. But look at those 6 actions, right? Give every child the best start in life, enable all children to maximize their capabilities, create fair employment, ensure healthy standards of living, create and develop healthy and sustainable places and communities, strengthen the role and impact of ill health prevention. The first 5 don't even mention healthcare. They don't mention doctors, they don't mention nurses, they don't mention medicines, right? So this is a really important point, that health inequalities, what he was saying was health inequalities is not the concern of just healthcare people, that it is a wider issue of society. Yes, we've got our health prevention piece here, critical part in public health and health promotion, but all of these are what comes first. Now, the next couple of slides, I have all his messages of the Marmot Review. That's a bit too much, I won't go through them, but take my word for it that they don't focus on hospitals and beds and healthcare systems. They focus instead on the community. I was lucky enough to meet him at a summer school that was on the social determinants of health, and I got to chat to him briefly. And when I brought up Ireland and working in Ireland and the health system is different, he was like, no, don't even mention the health system. It's not about the health system. It's just not. And so after he gave me a good ticking off, he went on to tell me about the wider perspective. And I think I finally got the message then. So these are social determinants of health. It is about housing. It is about access to green spaces. It is about transport. It is about early childhood education. It's this wider, these conditions in which people are born, grow, live, work, and age, including the health system, but not exclusively. So it's the wider issue. I've mentioned already Wilkinson and Pickett. So Marmot did such a great job on the UK that he went on to bigger and better things and got involved in the World Health Organization's Commission on the Social Determinants of Health. So he took his work and did a global review, and the Commission on the Social Determinants of Health in 2000 published 3 overarching recommendations. It's a document about this thick. If you get a chance to look at it, it is interesting. But the 3 main overarching recommendations were: improve daily living conditions, tackle the inequitable distribution of power, money, and resources, measure and understand the problems, and assess the impact of action. So again, it's not all about necessarily healthcare. I'm going to skip over that one and just give a shout out for nurses. So he also said, Sir Michael Marmot, you can see I kind of adore the guy, but he also said that nurses are the most trusted group, as the most trusted group of people in healthcare, and rightly so. They treat individuals with compassion and care and have great potential to improve the health of communities through action on the social determinants of health. So he can see that this is an issue for all of us. So what am I getting at? There's numerous reports. We've known about this for some time. There's lots of evidence around this. In Ireland specifically, we know we have a deeply inequitable 2-tiered health system. Some of you might recognise that photograph. That's a photograph from when, a number of years ago, when the government suggested that they might remove the health card, what am I trying to say, the medical card from the older adults, and they rightly protested and got out on the street, and guess what, they changed their minds about that. But we do have a deeply inequitable healthcare system, and for some reason we've kind of allowed this to continue in Ireland. And my PhD was looking at what had happened in the past to work out why this had happened. I won't start talking about my PhD, don't worry, but it was interesting to look at. And we know that the health service does influence health inequities. And we also know of what's called the inverse care law, which I know some of you will have come across before, which describes, and we've known about this since 1970s, that health services are of poorer quality and less accessible in the disadvantaged populations that actually need them most. And we have an inverse true as well. So in areas that are least in need, there are the most services. So we have a very strange system there. I'm thinking I better get a move on and get on to COVID here, hadn't I? I'm running out of time. Okay, so access to health services is about more than just getting to the hospital. It's about larger barriers in equity and access, cultural barriers, language barriers, all of these. I'm going to move on to COVID. And very briefly to finish up, we know that COVID has lifted the lid on a lot of these inequalities and has magnified them for everyone to see. So things that we may not have been able to see. We know, for example, from this study that COVID-19 has shown around respiratory health inequalities. There's evidence being published all the time now around the social determinants of health and inequalities. And we know that we need governments to take actions to mitigate the various negative effects of protected and vulnerable groups, especially considering what policies they could bring in to alleviate the economic impact on the very low income earners, and the impact that we know that this has on healthcare. A phrase that has been coined recently is a syndemic, that COVID-19 isn't just a pandemic, but is a syndemic. A syndemic or synergistic epidemic is the aggregation of two or more concurrent or sequential epidemics or disease clusters in a population with biological interactions which exacerbate the prognosis and burden of disease. So Horton, who references on the last slide, describes how COVID-19 clusters with pre-existing conditions, interacts with them, and is driven by larger political, economic, and social factors. More evidence around increasing multimorbidity. We know people who have cardiometabolic multimorbidity and polypharmacy. We know that these people are at higher risk as well of developing COVID-19. We also know that this intersects with other additional factors such as non-white ethnicity, and that people are at heightened risk then of COVID-19. So those with multimorbidity, twice the infection rate of COVID-19. Finally, a quick mention of intersectionality, which I promised. This is when multiple oppressions and their impact, where class and ethnicity, gender and class overlap. So for example, class and ethnicity or gender and class might overlap, that people face deeper and more complex forms of inequality. So that's the key words, that they're deeper inequalities and they're complex. So that means they're difficult to solve. And that these diverse social inequalities intertwine in social life. And it also means that while they intertwine, one solution for one doesn't necessarily fit them all. So lots of evidence has been formed around the fact that we know that COVID-19 has raised our awareness of the intersectionality of various structural inequalities. I should really have a conclusion slide. I've just realized I don't have a conclusion slide. What sort of lecture is she? I don't have a conclusion slide, but I do have further reading for you. And I hope that was of interest. And I suppose the take-home message would be that the issue of social determinants of health and the inequalities that we know exist in health are wider than the remit of the health service, and that it is a wider sort of all of government approach that is needed to address it. And I'm going to leave it at that.
Angela, thank you very much. That was very insightful. I mean, I never realised that there is such a stark difference in mortality between socioeconomic groups, you know, in places like the UK. You would have an awareness that it's like that worldwide, but maybe not so close to home. So thank you, Angela. It was very insightful. So we'll take a few minutes for a few questions, if people have questions now, or possibly we can take them at the end. If not, but we have one question here from Doris Whelan, who asks, he thanks you for your presentation, and then he asks if you could comment on whether sláinte care will make much difference. I suppose that's to the inequalities that we see in Ireland.
Yeah, it's a really good question, Darius, thanks. Yeah, I mean, the ambition of sláinte care is to deal with the issue of access to healthcare. access, getting those appointments, getting those initial appointments with consultants of speciality, for example. And that's critically important. And it will help. But I would say that given that we know that the wider impacts of structural inequalities and their impacts on health, that we certainly need more than staunch care. And we need to be looking more widely at things like our transport system, at our infrastructure for cycling, safe cycling. We need to be looking at early childhood education, preschool, and all of that, given that we know that this is a cradle to the grave situation. So it will help, and it's certainly urgent, but it won't be the full solution.
Yeah, thank you, Angela. So we don't have any other questions coming through at the moment, but maybe if people do have questions, we could take them at the end after Dr. MacSharry speaks. So thank you very much, Angela.
Thank you.
So Anton, I'll hand over to you then, if you'd like to introduce Dr. MacSharry.
Yeah, of course. I'll just quickly share this one slide. So Dr. MacSharry graduated with a Bachelor of Science in Microbiology and a PhD in Mucosal Immunology from the University College Cork. He worked with Elementary Health Limited as a molecular biology section head, collaborating with several multinational research partners. In 2008, he joined the APC Host Response Corps as postdoctoral researcher and collaborated on research with GlaxoSmithKline. Dr. MacSharry was a guest lecturer at the Meekins Christie Laboratory, McGill University in Montreal, Canada, as part of an SFI fellowship in 2009 and 2010. Dr. MacSharry has also lectured and supervised part-time in the CIT. Dr. MacSharry was recently appointed as a lecturer in the molecular microbiology and assistant director of the graduate entry medicine program with the School of Medicine with an affiliation to the School of Microbiology. His research interests are in host microbe interactions with particular focus on the immune sampling and response in the gut and the lungs. And so with that, I can hand it over to Dr. MacSharry.
Thanks very much, Anton. So I'll just share my screen. So thanks, Anton. I'd say that was an old enough biography, so it's making me feel younger. So I'm just going to talk to you today a small bit about using saliva to detect SARS-CoV-2, and I think it's very pertinent, especially what Angela was talking about earlier about the community and trying to provide healthcare, but also giving people access. And so hopefully you'll see my point at the end of how we can use this to kind of, I suppose, track down and eradicate SARS-CoV-2 or COVID-19. So again, so this is just your general introduction slide. The numbers keep on changing. So we're at 2.5 million deaths now at the moment, and there's around 130 million cases. Obviously, North America, India, and Europe are the main spots really, and obviously the more virus spread, the more replication of the virus, the more mutations, and this will change then the terms that we've all gotten used to hearing of R0 and quarantine, which is from our Phoenician friends from a few hundred years ago, quarantino, and again the R0 value will change depending on the prevalence of the virus and the mutation. So at the moment, you'll be hearing a lot about the UK variant or the South African or the Brazilian variant, and it's becoming more infective and it's becoming more host adapted, and that's why the numbers aren't dropping as fast as what we're seeing since the spike. Maybe we had April and May last year, and it dropped way down, and now it's come back up and it's harder to drop down, and that's because of the transmissibility is increased. Okay, so I suppose back to the topic. So I suppose obviously SARS-CoV-2, how is it spread? It's aerosolized really by saliva droplets and respiratory inhalation. Obviously we're all doing a lot of alcohol gels and soaps and hand washing, and I suppose closing toilet seat lids and not shaking hands, but in principle really it is spread by respiratory droplets, and that's why mask wearing is effective. And this is a lovely image from Niall Smith from CIT here, just showing the vaporization of when you're breathing, and how a mask will reduce the vapors, not prevent it completely, but it will reduce the droplets. And that's why mask wearing is essential. fomites and alcohol gels probably have a role, but they're good for sanitation, but it's the masks that are really going to reduce the spread. So again, so just a bit of this is a bit of medical microbiology that all my poor medical students have to listen to me. So the virus has the target to a receptor, and we all have heard of ACE2, and it's a common enough receptor associated with the blood pressure, regulation of the blood pressure system. And again, I just highlighted some of the key areas where SARS-CoV-2 infects, but it can infect throughout the body because the ACE2 is associated with lots of these sites because it binds, it's an epithelial cell receptor, and it regulates the blood pressure. And then obviously with SARS-CoV-2, when it does infect, it targets your epithelia and gets down into the lower respiratory tree, and that's where it causes a lot of cell damage and death, and then you get hyperinflammation and a lot of fluid on the lungs. And this is the big problem with SARS-CoV-2 when it does progress down to the lower respiratory tree, is the fluid and the inflammation, and then your body goes into distress and you can't breathe. And that's why some people are on positive pressure oxygen or intubated. Again, so this is a key slide then for, as a medical microbiologist, all my Gem 1s will be familiar with this receptor need to bind, but you also need to realize that it needs a serine protease to activate it to get in. Okay, and then what happens is then we have the virus, and it has a kind of a low level of replication before we start spotting the virus and the symptoms. And then eventually then our immune system will clear the virus, and we will develop an immune response or antibody response to it. However, some people do progress on to hospitalization, and which can, be more have a mortality issue as well. So, sorry, let's get my slide to change. It won't change. Sorry. So, 2 seconds. Yeah, here we go. So, again, so how do you detect the COVID-19 or SARS-CoV-2? So, in the clinic or in the hospital, it's using a nasopharyngeal swab. And what they're doing is they're going up through your nose and they're swabbing the back of your respiratory tract, your upper respiratory tract, and they're targeting the epithelial cells that are where the virus is replicating. And that swab then is put into either a virus transport medium or a lysis buffer, which breaks open the virus. And then the RNA is purified and converted into cDNA and then amplified into PCR. And these are some amplification plots that you'll see. And basically, the more RNA or cDNA that is present, the earlier the cycle number. So you'll hear of early CT and late CT. And late CT is at the very end of the detection here. And this is where we would be always dubious when you find a very late CT, because sometimes you do get random events in the PCR cycle. But generally, patients that are in hospital and do detect with SARS-CoV by nasopharyngeal swab are usually up around here at the very early CTs. And this is because they're shedding so much virus, because the virus is actively replicating and is present. Now, obviously, when you swab here in the nasopharyngeal, you're swabbing the epithelia and the upper respiratory tract. And our question in, I suppose it was April and May last year, was because we do a lot of respiratory microbiome studies in the lung washes and saliva of asthma patients, was what about saliva could be useful rather than somebody PPEing up to take a nasopharyngeal swab. You could just give a saliva sample because this ACE2 receptor is expressed in the epithelia, especially around the salivary gland. So that was kind of our thoughts. And so we also then, did a bit of research, and some of us have had experience of getting a nasopharyngeal swab. And a nice way of saying it's like your brain's been tickled via your nose, or a brain biopsy. And you get some humorous stuff on Twitter about it, how you feel when you get your swab. And then some people actually have, encaphocele, which are just a kind of I suppose a hereditary, malformation, and they can lyse actually, if possible. So that, you could serve with cerebrospinal fluid leak, which is not a nice thing to have. So obviously it's a quite invasive procedure. And I suppose one of the big things that we think as well that was at the time was that, PPE was scarce, healthcare workers were getting sick, and that, if you could self-sample rather than having somebody sample you, it would really free up the healthcare workers, especially the nurses and the doctors, to be able to, help patients who were sick with SARS-CoV-2. And at around the same time as when we were talking about this, SFI released a rapid response grant call. Sorry, skip to a rapid response grant call. And what they found is that they said, right, come up with new methods or novel techniques to help track SARS-CoV-2. And so we just started putting together a plan for saliva as a method of sampling. At the same time as well, somebody published a paper showing the linkage between your salivary glands in the back of your upper respiratory tract, that there's new ones called tubarial salivary glands, which are also linked. And this is actually the site of where the nasopharyngeal swab is. So basically we're sampling saliva by MPS anyway. So then we put together our proposal on saliva using these preservative tubes, with the aim of the preservative tube, because a preservative tube would be useful in the fact that you could inactivate the virus, so you could use it for a community sampling. So for instance, we could send it out to a school, we could send it to, some, like as Angela was saying, you might have some certain communities maybe disadvantaged. You could provide them with saliva tubes where they'd self-sample, and then we'd bring it to the lab and process it. So that was the aim. And again, just to divert for a small bit, saliva and microbiology, and obviously as a microbiologist, the godfather of microbiology is a guy called Antony van Leeuwenhoek. He's a Dutchman, and he used to make these tiny little lenses of 2 millimeters in the 1600s. And there's Anton with his lab supervisor there. You can see, look, gender equality. And he basically sampled his saliva, and he was the first person to ever see submicroscopic, in other words, stuff that our eyes couldn't see. And he was able to see bacteria and parasites and amoeba, and he coined the phrase called animalcules, which eventually became microbes because of the microscope. And this is the microscope he was using. And so again, this was the kind of the birth then of microbiology in detecting these pathogens that cause illnesses. Prior to this, it was the theory of spontaneous generation, or miasma, where basically we were doomed by a curse and we picked up a disease. So it kind of changed human perception of disease. And it's obviously where we're at now, where we can find SARS-CoV-2 by, not microscopy, but even molecular techniques going down to nucleic acids. So back anyway to the saliva sampling. So again, saliva sampling is quite easy to do. You can just spit into a tube. You can do a saliva swab around your mouth. There's lots of collection devices. It's common enough in the medical field where you have swabs or pops, they call them. And it's quite a useful way for looking for hormones and cytokines. And obviously, as a microbiologist, there's lots of microbes in your mouth. And that's why we all visit a dentist. And there's probably more microbes in your mouth than human cells. And this is the plaque that forms in our teeth. So we thought it'd be a very easy way to, to look for SARS-CoV-2. And then we could use our standard RT-PCR, and then also use kind of some newer molecular techniques and to look for the viral genome. So we put together our grant. SFI put the call out in April, and the deadline was the 1st of May. So we put together our grant, and I suppose you break all your scientific projects down into work packages or tasks, which is brilliant. And so our first task was we wanted to compare the saliva versus nasopharyngeal swabs. We also wanted to look if we could kill the virus with these preservative tubes, because if the virus was so resistant to these preservatives, you might be spreading the disease in the community. And one of the things we noticed when we were, you saw in the media, there's a lot of hysteria about SARS-CoV-2, and some of the scientific principles were being ignored. Detergents break open lipids, alcohol dissolves lipids, and they're kind of scientific facts, but a lot of this was being ignored. And so we said, right, well, we'll prove that these detergents can kill the SARS-CoV-2, because they've been proven to kill Mycobacterium tuberculosis, but no one had studied SARS-CoV-2, so we couldn't get permission to handle the samples unless they were inactivated. So we said, right, we'll prove it. So we, in collaboration with some colleagues, we went out and designed that package. We also said saliva could be useful for checking the shedding of the virus. So when you turn up to the hospital and you're positive, how long are you shedding that virus after you present? So can you be released from the hospital? Do you get better, but do you still shed the virus? All these questions that no one really knows. And then we wanted to use some of these rapid detection mechanisms that you hear about in the media, which give you really fast answers, okay? And I suppose the big thing, and I mentioned it again, is that saliva sampling reduces the need for medical staff to take samples. So people are freed up to work, they don't need to be PP'd up. And in our method with this DNA Genotech company, it inactivates the virus. So then you don't have to worry about having an infective sample being sent by post or in somebody's courier, which is, a crucial thing to figure out. Okay, so we found out in August, then the start of August, that we got funded, which was great. And then we had set up networks in the meantime where we were able to get samples. And sorry, my slides just take a second to change. So it's just giving me an alert, 1 second. And yeah, here we go. So sorry. So this is kind of just a breakdown of the tasks, and we were able to collect saliva samples from healthcare workers in CUH, and then we started talking to colleagues like Cillian de Gascon and some colleagues in Dublin like Kingston Mills. And we also, a lady called Virginie Gauthier was able to show us that the virus was killed by these preservative tubes. So we pulled all this project together, and so we're still analyzing, we're still processing some of the data, and these are some of the colleagues that we all met up. And this is one of the good things with COVID, is that usually you're applying for grants for research, you're probably competing against everyone, and but with SARS-CoV-2, it kind of broke down the barrier, and we all just wanted to help the healthcare workers and help the medical scientists. Some of us made lysis buffers, and we put these grants together, and we kind of all linked up networks together. So we had a network with Professor Liam Fanning, Professor Mike Prentice, Professor Mary Horgan, and Korean Sadler, 2 infectious disease consultants. And we linked up with Andy O'Connor and Tala, and Cillian de Gascon in the NVRL, and Sinead Kaur, and Liam Gallagher, and Virginie Gauthier, who's a virologist who studies HIV, and Kate Reddington in Galway. And we all put together this project, and we got virus samples. And then we were able to pull all these together, and with Cillian de Gascon, we were able to also compare saliva to nasal swabs, which are used sometimes in children, because sometimes kids can't give you saliva. And obviously nasopharyngeal swabs are very hard to do in children because of their anatomically structured, that is, they're kind of short, that it can be fairly traumatic for them. So so we pulled this together, and we were looking quite good. So far, it looks like saliva is giving us as good as the nasopharyngeal swab. There are 1 or 2 issues, but that's always the way, and I'll discuss in a second. So at the same time, we weren't the only ones thinking of this. Anne Wiley and her group in Harvard also developed a thing called Saliva Direct, where they actually just take saliva, dissolve it with an enzyme, heat it up, and then they compared it to nasopharyngeal swabs. And they found basically that they were as comparable. And this is published in the New England Journal, which is fairly prestigious, in September. And so they were able to show that you could detect the saliva without using the need for swabs. There was also reports coming out in February from a Chinese group by To et al. Showing saliva presence in Hong Kong in some of the saliva in the SARS-CoV-2 patients. So I suppose these are just some highlights, what they say. So higher saliva versus NP swab. So in other words, probably a higher viral. Now, again, it depends on the kinetics of how the virus is shed and how someone is infected. But basically, it showed that saliva is as good as or as sensitive as the nasopharyngeal swab, which was reassuring to us, because when we designed it, we were going, okay, hopefully it works. And then in the last few months, there's been lots of preprints, which you've seen if you look up in the media, you'll see a lot of things called preprints, which are these people put up their papers online in things called medXRIV or BioRIV. And before they've been peer reviewed, but they're basically being submitted for peer review. And we found the Saliva Direct, which is the Anne Wiley and our colleagues in Harvard, where they commissioned with the NBA, the basketball in the US, where they sampled everyone with saliva sampling, and all the players were sampled. So if there was any detection of SARS-CoV-2, they were quarantined. And and then also in Illinois as well, the university, they also developed a method where they just heated up the saliva and put in a detergent, and they were able to find the virus. And then there was these rapid testing, which were done in Trinity. And you'll see these are the same tests that you'll see in the airport tests, this rapid colorimetric test called LAMP test. And it gives you a yes, no by color. And these were set up in Trinity College and also in the airports. And these are quite good as well. And then you'll hear of another one, which is a fluorescent made called CRISPR. And all of these were just showing that basically saliva is useful for detecting the virus. And because of all this, we all started talking, all the different universities. McLean Farley, who's in Trinity College, set up the Saliva Working Group, and in conjunction with Kingston Mills and Charlie's Plan in Galway and Liam Gallagher in UCD, we were talking to SFI about the use for saliva in screening for SARS-CoV-2. Apart from just using your typical RT-PCR, you can use these new techniques such as the LAMP technique, which is a rapid 30-minute test. And then you can send these samples off for sequencing to look for the variants that are present, okay? And then we also discussed maybe the future of looking for antibodies in saliva to look for, the evidence of vaccination or infection. Now, we did discuss there's always issues and there's always problems in science. We did find with the LAMP assay that it was sensitive to the pH of someone's saliva. So sometimes when you take a saliva sample, you ask people to refrain from eating for roughly 30 minutes or drinking coffee or tea, especially coffee because it's quite acidic. And obviously, there's issues, and sometimes people will give you more than just saliva, they'll give you sputum from the lower respiratory tract, which can confound the samples as well. But we did find these are some limitations to the sampling, but again, we found it very effective, and there's different groups around Ireland. So we've just talking to Mark Ferguson, SFI, yesterday about this. So hopefully it might be utilised more widespread. So I suppose this is just kind of synopsizing really the role for saliva and COVID. Obviously, depending on your viral load. So usually your viral load is very high, when you've gone through the pre-symptomatic and you're in the active infection. And then sometimes your viral load will drop off, and the different tests that you'll hear about. So you may have heard of antigen tests in the media, and the UK government are buying in millions of them to spread around the UK to look for the virus. These do work, but you need a high viral load. So you need to be kind of basically in the middle of, the highest infective phase of the infection. They take about 20 minutes. They're very easy to do, just like a pregnancy test. They run on a kind of a membrane paper. There's another assay called the LAMP assay. It's a 30-minute, and that's the colorimetric one I showed you previously. It doesn't require much equipment except a heating block, which is quite easy to do. And then your standard, which you've all heard of, is RT-PCR. The PCR requires, robots for extraction. It requires, fairly big pieces of machinery that give you the analysis. So this is why usually it takes roughly 24 hours, while some of these tests here are a few minutes. And, so again, but they do have, the antigen tests sensitivity wise. We did some tests in our lab with Liam Fanning and myself, and we found that it can't detect 10,000 viral particles. So, basically if you've got a low viral level, or you might be at the part of the curve here where you're just starting the pre-symptomatic, or you're coming out the end, you might be a negative when you could still be there, but depending what assay you use. Okay, so again, so you might use saliva, but you're depending what assays you're using. So this is kind of how I've ranked them, but also these are more expensive as well the farther down you go. So I suppose then I'm nearly finished, but so I suppose there is a need for saliva, I think, now in COVID-19 testing and screening. This is just a headline I saw today on the Irish Times. NPHET says normal life is some way off and vaccination unlikely to fully halt it. Now that's probably because of the variants, but also the fact that what we need to do is we start need to looking more in our community. We don't need to be waiting for someone to turn up. And I know some people won't turn up and say that they're sick because they're afraid they mightn't get paid the next day if they're sick, and that's something to consider. And so what we propose is sending out these saliva testing tubes or saliva sampling stations where people can just spit into a tube, drop it in the post, it's inactivated, and we can give you an answer, or you can go to a saliva station and we can do a really fast test or a LAMP test or even a PCR. And then we'll be able to find these, we hear about super spreaders spreading events, or someone's asymptomatic and they turn up and they're tested and they have no symptoms. And the only way you're going to find these is if you check the whole community. If we're waiting for people to present, we're never going to track the virus. And that's why we're in this lockdown, because, the prevalence of your virus depends on how much time you're looking in the community. So if you don't look and you don't find, your prevalence is skewed. So we think saliva will be a really useful way to find where the virus is in the community, isolate people until the virus clears, and then reduce this. And again, it's aerosolized. And again, what we're finding, especially in the in the older healthcare system, the old folks' homes, is that you know, your immune system starts reducing, and you probably get chronically infected and shed. And the best way to go to those guys is a saliva test or a swab, and to look for it and see where it is, rather than waiting until they present in the hospital and spreading infection throughout the hospital. And again, you know, it's the same with the meat factory workers. These guys are on contracts. They don't turn up, they don't get paid. And I know the Department of Agriculture are screening their staff as well. And we have sports teams such as the rugby teams that are doing it. But I think as a community in Ireland, and this touches on what Angela talked about, we need to be saying, right, we need to do more and more community screening, and so that we can find exactly where the virus is, because then you can know, okay, it's here, we didn't even think it was here. And that's how you're going to find it. You can't wait for people to keep on reporting, because you'll never stop it. And you can blame people traveling, you can blame different groups, but you have to put your hand up and say, we need to screen more. And that's kind of my rant, really. It's the headline set me off. And so then just the last slide, again, another way to check as well for the presence of SARS-CoV-2 is to look for the community wastewater screening. And so you can take wastewater sampling, and you can find the virus there, and you know then it's in the community, even if no one's reporting. And so Dr. Niall O'Leary here, microbiology, was funded in collaboration with Liam Fanning and myself, where we're actually looking for the SARS-CoV in the water samples. And Niall is sampling around Munster, and we're finding it at low levels but present in the wastewater. And you know, so that's showing that the virus is present in the community. And if you think about, in Ireland it rains a lot, so there's a good dilution effect. So most of the wastewater is fairly well diluted by the time Niall is sampling it from a river or an outfall. So the fact that we're able to detect it by RT-PCR, and that the virus is sensitive to lots of issues, it means that the virus is there and it's being shed into the community, and it needs to be, you know, so it needs to be monitored. And then I just want to give a shout out to, we had a final year student who worked with Niall last summer, and it was Leah McPhillips, and she was doing a project on saliva RNA detection with me for a final year project before Christmas, and she's able to detect down to 6 virus particles by PCR in the lab. So again, you know, the techniques work, and I think if we do more community screening, be it saliva or wastewater or swabbing, we'll find the virus. And I think with that, I'll sign off. So thanks very much.
Thank you so much. That was really, really interesting. And I guess now we can open the floor to any questions. So if there are any questions, you can either write in the chat or feel free to unmute yourself and ask away.
Dr. MacSharry, I might just ask one question there while we're waiting for questions to come through. You said that you detected the virus in wastewater. So what would the likelihood of it, like how likely would you be then to pick the virus up from, you know, if you're swimming in the sea or that?
Well, you know, I think there's definitely a chance. You know, it's, you know, if we're detecting it, when you think about the dilution effect of what Niall is doing, you know, yeah, they're just big water samples, but you know, if it's detectable, it must be there. Whether it's infectious, what we need to do is send it off, try and culture it in labs. You know, we have to have a level 3 lab to culture the SARS-CoV-2. So to do that, at the moment in UCC, we have to get more funding to get the lab up and running. But our collaborators, you know, said that they could look for it. But the fact that you can find the virus, the nucleic acid is very sensitive to degradation, so that it's there at all, it must mean that it's a viable virus.
Yeah, well, I didn't think that that you'd be picking it up in that kind of situation, you know. But
yeah, well, like a lot of people pick up E. coli. You get these you know, you hear of a lot of surfers in Cornwall, and you know, it's the same in Ireland as well. You know, if there's a big rainfall, you get a lot of flushing of E. coli and slurry from the farms into the rivers, and then they come out near the nice surf spots, and a lot of people pick up surfers' throat or kind of they get E. coli and stuff. So yeah, it's definitely a possibility. I think it's something that needs further investigation. Niall's doing great work on it.
Yeah, John Kenny asks, is the wastewater testing being used as an early warning at the moment?
It isn't being at the moment, but it would be great. I think, you know, if you could go sampling around and say, right, look, it's in the community, we know it's wherever it is. Obviously, Irish Water will have their protocols as well for treating the water. But you know, I think it would be very useful to say, okay, we can detect it. You know, maybe it's too low a level to cause infection, as you said, Kate, but I think it should be, you know, employed. You know, the only way we're going to stop this virus is the community. I know the focus is to keep the hospitals running and not swamped. However, it's never going to go away unless you find it in the community.
Yeah. There doesn't seem to be any other questions coming through there. But we have
actually, I can ask a question.
Yeah.
Yeah. So I was just wondering, how long could the viral particles survive in the saliva test kits? Like in cases, for example, where you know, the I guess testing services are at maximum capacity. Like, would it be able to just people just hold on to them and test them later, or like?
Yeah, so I think talking depends on what you take the saliva sample in. So you can put it into just a tube, you know, spit into a tube, and if you put it at 4 degrees, the virus is probably still detectable for a few days. Once it's stabilized, you know, you just want to reduce the breakdown of the viral coat. But what they're doing a lot with the nasopharyngeal swabs now is they do the swab, and it goes straight into a detergent. So then they get nucleic acid straight away. Our saliva tube system that we're using, there was 2 reasons really. Was that one, to preserve inactivate the virus so we wouldn't get in trouble with the safety authority guys, and but that preserves it forever then. So the tubes we're using, you know, they can be stable at 37 degrees for, you know, a month. So you'll find a nucleic acid, but in theory, you do get false positives and false negatives as well, you know, with any test, the nasopharyngeal swab to, you know, saliva, to any of them, because, you know, there's always a chance that something isn't done correctly or it isn't handled. And killing the gas gun was saying one of the big issues with the you know, the testing is that getting the sample from the patient to the actual lab. You know, the lab can run it really fast, but it's getting it from the patient to the lab because you need to transport it, and you know, and that can reduce your sensitivity as well.
Right. Thank you. And also, Kevin asks, would there be a lot of viral debris in saliva samples compared to NPSs?
I suppose that, yeah. Well, once the virus is shedding, once the virus was replicating and shedding in the epithelium, you will get viral particles. Whether there's much viral debris, you know, you might get the viral debris would be assuming that the virus is being lysed in the saliva. But I don't think it will lyse too much in the saliva because it seems to be aerosolized quite easily. So it seems to be resistant to lysis in the saliva. But so I think one of the big issues with saliva that may be a confounding factor is how soon after eating or drinking that you can, you know, when someone gives you saliva, are you getting an expectoration, or are you just getting a saliva just from the mouth? The viral debris is usually when they're lysed, so I don't know how much the virus will lyse. You know, it's probably ineffective for it to lyse that fast.
Thank you, John. So I think that's all the questions that we have. So just to extend our thanks again on behalf of both the societies, to both John and Angela for giving up your time and coming to speak to us this evening. It really was fascinating and so interesting to see how, you know, the science that you've been developing, John, links into the health inequalities that Angela spoke about. And yeah, you know, fingers crossed that we start to see more widespread testing, because, you know, I think it's the only way that we'll really be able to get back to normality that we all really need at this stage. So thank you guys, and thanks to everyone who came and listened to us this evening as well. You know, it wouldn't be an event without the attendees, and of course thanks to the Biomed and MRT and Pharma societies who were involved in the COVID conference as well. And there's another final instalment tomorrow night hosted by the Biomed Society, which I'm sure will be another fantastic evening of interesting speakers. So so thank you guys, and I'll leave you go back to your families and back to your evening now. So
thanks for having us.